study embryo development, so we want to understand how a multicellular organism forms from a single cell, the fertilized egg. This is a really fascinating process, and we use model organisms to study this. And in this video you can see a developing chick embryo, which initially is a very, very simple structure, and over time becomes more complex and more recognizable features develop. Initially in this embryo, these cells are also very simple and similar to each other, and over time gradually then we get cell differentiation happening and different cell types appear. For example, heart cells appear, skeletal muscle cells appear, liver cells, blood, and so forth. I have always been fascinated how this happens. So how do cells become different from one another? How is this controlled? And it turns out that they send signals to one another, secreted molecules with which they communicate. And cells respond to that to decide their fate and to decide which path to follow. So similar to you and me being influenced by friends and family when it comes to important decisions in our lives, cells also receive their signals from the environment. They listen to extrinsic cues. And in the embryo, skeletal muscle develops from somites. Somites are paired structures which flow in the head to tail direction in vertebrate embryos to give rise to myofibers, which are illustrated here on the screen in green. And they are repeated units which form along the main body axis, and they give rise to much of the musculoskeletal system, including skeletal muscle, bone, cartilage, and tendons and ligaments, and so forth. And we can still recognize the repetitive nature of these somites in the adult body plan, for example when we look at our vertebral column or the ribs, which are also somite derived. My studies identified which factors are important for skeletal muscle development. This is important for stem cell biology and regenerative medicine, for example, where we want to harness this knowledge to help repair diseased muscle. Essentially, we want to use the embryo's own mechanisms to repair muscle that are used normally to build the muscle in the first place. This might also be useful to develop therapies to treat people that have been ill for a long time and suffer from muscle wasting, or for example in elderly people where there's also often muscle wasting that can lead to increased frailty.